Hello, my name is Justin. Thank you for the introduction. And the work I'm presenting today is about MOLIERE, a hypothesis generation system that I've been developing over the course of the last year with my advisor, Dr. Safro, and collaborators in the medical world, particularly Michael Sutman. So before I get too deep into what we're doing here, I want to give a background on hypothesis generation. I've noticed a lot of people might not know exactly what we mean by this term. So the general concept is trying to identify potential new connections within the set of already existing public information. And this idea was initially proposed by Don Swanson in the mid-'80s, and he believed that the pace of information had simply grown to a rate faster than any human being can keep up with. The set of public information is too large, and very often scientists will work on the same problem but from different directions. They might not use the same words to talk about the same thing, and they're probably not reading each other's papers or talking to each other. So a computer system on the opposite end of this can read everything and hopefully find these connections for us. Now, he thought about this in the mid-'80s. That's only gotten truer today. Right now, Medline, a data set that I'm going to be talking about more later, one of the main repositories for mostly medical information, is growing by upwards of I've heard this number as high as 10,000 papers daily. There are a lot more people in science. They're writing lots and lots of papers, and as a result, people are specializing more due to the demand of the field. This kind of increases the bias present within science. When you sit down to do a literature review, you'll probably read the people that show up on Google Scholar and the people that you're aware of or published at a recent conference. And you're not going to hear about the people working on your problem from different angles. So what we're going to attempt to do is generate hypotheses, but in a computational manner. So staying very basic, we're going to talk about hypotheses as if they are conceptually connections between entities. And this applies very well to the medical domain. It's one of the reasons we're here, where we might have a drug or an active ingredient, and we want to know what that drug might act upon to lower some symptom or cure some disease. And I'm going to make a caveat here that a system that I'm talking about in many hypothesis generation systems, although they would apply to many domains such as patents or economics, tend not to apply well to concepts like mathematics, where I can't really say that fractions have any relation to integration or something like that. So with this in mind, we're going to talk about an automatic hypothesis generation system at an extremely high level. The system that I'm about to describe is going to start with not just a small set of connections, but any sort of potential connection that we could find related to the active ingredient or disease at hand, but not just one particular query. We're going to generate the entire network of potential implicit connections. And once we've got a network, we can do network things, such as find a path in a graph. And that path might be able to tell us some information, but more importantly, it can point to the locations within the graph that we can extract some of the most meaningful information. So extremely high level, this is where I'm going towards, but before I delve into my approach, I want to give you a sense of the foundation we're building upon with 3 systems. It's a very small subset of what's out there that showed kind of different properties. So Aerosmith was initially built by Swanson, and this project, I believe, was finally published towards the late 90s, and they were actually able to find some very interesting results using, in this perspective, relatively basic text mining techniques. They were simply looking at how words co-associate within document sets. And that has the ability to find, for example, this fish oil is related to Raynaud's disease. That is something the medical profession was not aware of until a data scientist informed them about it. And this was actually then found years after Don Swanson did his text mining. Now, Disease Connect takes a different approach. It, instead of kind of looking at I'm going to say macroscopic association between documents, is going to look at how words are related to each other by looking at subject-verb-object relationships within sentences and use some medical-specific information to build small subgraphs related to genes and diseases. So the idea here is that a human being can look at a representation of a small subnetwork, and that will help help them infer potential connections. Now, BioLDA takes a very different approach than the other two. It is not necessarily a network approach. It is a topic modeling approach that is able to incorporate quite a lot of medical-specific information in order to build high-quality biomedical topics. And they were able to find a connection between venlafaxine and HTR1A. These two objects are both related to depression and anxiety, but using the historical data they were working with, no one document actually used those terms together. So some of the other methods have trouble identifying that type of connection. And that example is particularly important because I'm going to be repeating this later in the talk, so keep that in the back of your head. But the limitation to all of these methods that I've been talking about is the scope of the information. So Arrowsmith requires that you know 2 keywords, and we're only going to consider documents that actually use those keywords, and we're not going to have the chance to explore the rest of medical terminology. Disease Connect limits itself to only a certain set of human curated terms. Now, it's a very large set comparatively, but it's going to be domain-specific to genes and disease discovery. And BioLDA, once again, requires lots of medical a priori knowledge in order to make its predictions. So what we're going to do is construct a very large knowledge network that uses practically every paper in Medline, every paper of substantial length that we can learn something about it, and we're going to identify those meaningful paths between objects of interest. And from those paths, we're going to extract clouds of abstracts, or subcorpi, which we can use to mine for important topics. So there's 2 main pipelines here. First, I'm going to talk about how we build the network, and then I'm going to talk about how we pull meaningful information out of the network. So without further ado, we're going to start with Medline, that data set that I mentioned earlier. It's got approximately 25 million documents, and after you throw away the ones that are too short, you end up with about 22 very high-quality publications. We have the abstracts and titles that we're going to deal with primarily here. And this is provided by the National Library of Medicine. And we can take that information and apply some typically standard text cleaning processes. The Specialist NLP toolset is a domain-specific text cleaner, but we've also applied the Natural Language Toolkit. That's just a Python utility. And we've been able to get machine representable text here. Then we apply a recent technology called topical phrase mining. This has been produced within the last 2 years at the University of Illinois, and it is able to identify not a computationally explosive set of n-grams, but instead n-grams with sufficient support that are backed by a topic modeling process that result in only the most meaningful n-grams. So conceptually, we're going to take the token asthma and the token attack, and when they occur together, we'll know to join them together to represent an asthma attack. And that token by itself kind of conveys more meaning than either of those 2 words conveyed separately. So now we're going to do a word embedding, and we choose to use fast text for this. Now, you probably are aware of word2vec. Fast text is a more recent implementation of the same technique, but it extends the idea to not only represent one word in a vector space based on its neighbors, but also, if that word is of sufficient length, to look for subwords present within the long, typically rarer token. Now, this is important because we've done n-gram mining, so some of our longer phrases are going to have very meaningful subwords in them. And also, medical text tends to be formulaic when they're talking about chemical names or some certain topics. So we are able to get a high-quality representation here, but of course, many word embedding methods could be used. And once we have an embedding for each n-gram, we can fit a centroid for each document. This gives us a real-valued way to represent the conceptual meaning behind a document at least an approximate level. Which is okay because we're going to be stitching those together in a nearest neighbors network. This means that 2 documents will be linked together if they are semantically similar. Now this is important because we're not just doing keyword matching. As I've noted before, word usage changes over time. For example, initial hypothesis generation papers, we're probably talking about literature-based discovery, but I might be talking about text mining, and we mean the same thing. And typically those words will have similar neighborhoods and convey a similar semantic purpose, so they would likely be represented in a similar location in our vector space. So, long story short, we've got a layer of abstracts, but that's not enough. We're also going to take the UMLS metathesaurus. This is medical-specific information, and I'll note the first time that we've really needed medical-specific information. This is 2 million keywords, and they come with a human-curated network on top of that. And we attach that to the abstract layer using TF-IDF to weight the edges, such that a keyword has a low weight to an abstract, meaning they're close together, if the abstract contains a statistically significant number of occurrences of that keyword. Now, in the paper that I'm presenting today, this is all we've done. In our current work, we're also extracting the 15 million n-grams present within that abstract layer to find connections that are not simply given to us in this human-curated section. Okay, so this is the network as a whole, where I mentioned edge weight approximately means distance between two topics, and all of these edges are normalized per category to 0, 1, so we can make some meaningful relationships between them. And then we're able to do some tuning to try to talk about maybe we want abstracts to be closer to each other on average, which becomes important when we talk about the query process. So we start with this knowledge network that I've been talking about, and we select 2 nodes of interest. Now, once again, in a real application domain, we might have one gene we care about, and we want to know every disease it might be related to. But for simplicity's sake, we've got an I and a J. And we want to find the shortest path between them. And as I've mentioned before, the way we've weighted this network, the shortest path between 2 keywords that are sufficiently far apart are this path is likely going to take advantage of this abstract layer. Now, this is important. If we just used connections within the keyword layer, we're simply talking about 2 keywords that are either conceptually similar or have been linked together by a human-curated edge, and that's not an interesting hypothesis. So our idea of an interesting hypothesis is going to be a path that includes abstracts. And once we've got a path, these, from my experience, have been between 4 to 8 nodes long. We will extract additional documents from along this path so we can eventually apply a topic modeling process. So we do this by taking, for each keyword, the set of the most relevant documents around that keyword. And for each pair of keywords, we're going to take the set of documents that share both keywords. And for each abstract, we're going to take a wide neighborhood around that abstract in the nearest neighbors network. So the mental image here is that if a human being were able to read the 15,000 papers that we've identified in the network, they should be able to tell you how I may or may not be related to J. So we've got a corpus. We can run topic modeling. We apply an off-the-shelf topic modeler, PLDA+, to quickly identify a set of topics, and that gives us some results. Here's a bit of a cartoonized version of these results. I'm only showing 3 of the 20 topics that we generated for this result, and currently we're experimenting with different numbers of topics. And I'm only showing a couple of results per topic. And as you can see, this occasionally raises more questions than it answers, so I'm going to be talking about how we can use these topics to infer potential connections in a second when we get to results. So we've got the venlafaxine versus HTR1A example that I mentioned at the beginning of the talk, but we're also going to explore a drug repurposing application that this system might be able to address. So venlafaxine is a generic drug that's known to treat depression and anxiety, and HTR1 and 2A are both genes that we know were related to depression and anxiety, but at the time of the data that we were using, 2009, there had been no strong connection between venlafaxine and either gene. So what we would like to identify is there a connection with one or the other? We run our process, we generate topic models, and here I'm describing how depression-related keywords and anxiety-related keywords occur per topic per query. So the orange bars represent the connection with HTR1A, The gray bars that you can't see are represented with HTR2A. And here we show the strong correlation with one gene and a very weak correlation with the others. So this shows us we're going in the right direction. Think of this as a sanity check. And looking back at that cartoon of topics, we see antidepressant drugs, but we also see the specific names of certain antidepressant drugs that are related to venlafaxine. We see parts of the brain that produce serotonin and other parts of the brain that are receptive to serotonin and, interestingly, are controlled by HTR1A. So we are seeing the relevant information occasionally rise to the top, and we see that there can be work to be done to make these topics more interpretable. But we're then exploring the drug repurposing application, where we want to look at a wide range of topics and see if we can find new applications for old drugs. So DDX3 is a protein that in, I believe, the mid-70s was found to be linked to how HIV spreads throughout the human body. And so drugs were created in order to target DDX3 to treat these HIV symptoms. But more recently, it has been found that DDX3 can also regulate how cancers spread throughout the body. That means that if you've already built a drug for HIV, you might be able to use the same drug on certain cancer patients. Drug companies are attempting to run queries like this a lot because it saves them a lot of money in R&D and gives them a new market. So we were working along with Michael Sutman, the medical researcher, on our paper, and he identified the vague topics or the vague hypotheses that we might want to identify. We actually ran a large number to give him a large data set that he could analyze on his own. And these are the trends that we were looking towards. And in these findings, we were expecting to see cell-cell adhesion, cell matrix adhesion, and WNT signaling pathways. These are the specific ways that DDX3 is related to the process of cancer metastasis, how cancer spreads throughout the body. Now, the medical reasoning behind this, once again done by my co-author, he looked at specific hypotheses that we were not aware of being interesting, but he knew with his expert knowledge that if we were onto something, we should find. And he found cell-cell adhesion and cell matrix adhesion had high support in key areas. WNT signaling pathways did not have specific support, although the concept of signaling pathways was definitely present. So we're in the right direction as far as drug repurposing applications might go. So as kind of a summarizing of the application domain, we can not only do drug repurposing, but we imagine this system can be retrained on data from multiple domains. In fact, this summer, I actually worked with hydrologists at Los Alamos National Lab, and we're developing a version of this for water resources research and trying to understand how interactions in topology might affect chemical compositions of water. And of course, coping with deadlines, because anything that gives us hypotheses faster will speed up the process of research in general. But I'm of the opinion that a system like this asks more questions than it necessarily answers, but I feel like we can only ask these questions now that we have a system to ask them on. So interpreting the results is particularly important. That means coming up with more semantically coherent topic models, and that might mean coming up with new network representations of our subquery, or like of our subcorpi. System verification is of course important. We've identified a couple of data sets that might be able to provide to us a wealth of hypotheses that we might be able to re-imply using our system. And of course, there are issues with tuning and making this a producible system that we might be able to give to a drug company where they want the new ideas that have shown up within the last month. What are they? So it's a lot of very interesting work, and work that I'm happy to get started on, of course. Our code is available online. Our initial data sets are available online. There is a web form where if you want to ask a query of us, you can, although due to some security constraints within Clemson, it's not going to run automatically yet. And I am definitely happy to answer any questions here or via email, of course. Thank you for your time.